The muscular system, the molecular basis of muscle contraction. There are three types of muscles, and they are skeleton, cardiac, and smooth. Contractility is a fundamental property of cells. Most of these contain essentially the contractile apparatus as found in the muscle cells. But in the muscle cells, a larger portion of the cell's resources are for contraction than other cell types. If you'll look at the diagram, it shows you the skeletal muscles that are attached to a bone. On the top, the cardiac muscle, which is in your heart, and the smooth muscle, like in your arteries and your intestines. So the skeletal muscles have contractions that move part of the skeleton, and they are voluntary. The apparatus is striated. The cardiac muscles make up the muscular walls in the heart, the myocardium. The cardiac cells are involuntary. It also has a striated or striped appearance due to the repeating sarcomeres. The smooth muscles are found in the walls of most blood vessels and tubular organs. The smooth muscles are involuntary with a smooth appearance as there are no striations. Contractile proteins are more randomly arranged in the smooth muscles than in your skeletal and cardiac muscles. Vertebrate skeletal muscles. So the skeletal muscle is also called a striated muscle. Each muscle fiber is a single cell that is packed with bundles of myofibrils made up of actin units surrounding thick myosin units. The sarcomere is a repeating unit made up of myofibrils of a muscle tissue. Z-lines border the sarcomere, which anchor the thin actin filaments. The I bands are only actin protein, while the A band is an actin and a myosin protein overlap. The A band contains all the myosin filaments. The H zone is the central sarcomere, and it is only myosin. The H zone and the I bands are areas where actin and myosin do not overlap, so they appear less dense here. So you can look at the diagram, and on the bottom, in the dark purple and then light purple colors, you can see the A band, which act on the actin and myosin. Then you'll see your I bands, another A band, and an I band. Up on the top, you can see the actin filaments, the myosin, your thick filaments. You can see where your Z lines are, your sarcomere. You can see that there is a Z line on both ends of it. And then you can see the sarcoplasmic reticulum, the yellow down there on the end, and then the skeletal muscle fiber around the outside of the fiber. The sliding filament model. So the sarcomere length is reduced while the Z line becomes shorter. Actin and myosin and their overlap increases as they slide past each other and the muscle contracts. The sarcomere shortens as the muscle contracts. The H zone and the I band become narrower. The Z lines move towards the A band. The actin filaments are sliding into the region occupied by the myosin filaments. So if you look at the diagram, you can see where the top one goes to the bottom one. This is what happens during a contraction and how they slide from the outside in. The actin-myosin interaction. The myosin head hydrolyzes ATP to ADP in the inorganic phosphate. This is called the high energy configuration. The myosin head will bind to the actin, and this is a cross bridge. Then releasing ADP and inorganic phosphate, the myosin relaxes the sliding actin. and this is called the low energy configuration. Binding of new ATP takes place, releasing the myosin head. The creatine phosphate is the supplier of phosphate to the ADP. The myosin-actin interaction also changes the physical properties of the mixture. So in the concentration of ions in the solution, if the concentration of ions in the solutions is low, myosin molecules aggregate into filaments, and as myosin and actin interact in the presence of ATP, they form a tight, compact gel mass. This process is called superprecipitation. If you look at the diagram, it starts with 1 at the top, and this is where the myosin head is bound to the ATP. This is part of the low energy configuration. You go around to 2. This is where the myosin head hydrolyzes ATP to ADP. Then you move around to 3, which is at the bottom. And this is where the myosin head binds to the actin, forming the cross bridge. And then continuing up, you will see the release of ADP, excuse me. And then the thin filament will move towards the center of the sarcomere. And then the binding of the new molecule of ATP releases the myosin head from the actin, and a new cycle will begin. Muscle contraction regulation. We're going to be talking about relaxation and contraction. So during relaxation, the tropomyosin blocks the myosin binding sites on actin. Then the contracting muscles take place when calcium binds to the troponin complex. Tropomyosin changes the shape of by exposing the myosin binding sites. Okay, the change in electrical properties of the transverse tubules during an action potential causes the rapid release of relatively large amounts of calcium ions into the sarcoplasm. And as the concentration increases, they become bound to the troponin in the thin filaments. And this removes the troponin and tryptomyosin mediated inhibition of the myosin-actin intervention. So while the muscle stimulation of an action potential is a motor neuron, it continues and calcium ions are being released. Some of the calcium ions are being removed from the sarcoplasm by the sarcoplasmic reticulum, the longitudinal tubules. The calcium ions are inside the tubules and they will slowly fuse out, and they are bound to a protein as a storage site. It requires energy for the removal of calcium ions, and the breakdown of ATP is the chemical reaction that supplies the energy. So for each ATP molecule that splits, 2 calcium ions are actually removed. Okay, and this is a muscle fiber. You can see the cross section. You can see where the action potential, where the yellow nerve binds to it. You can see the action potential off the motor neuron coming in. Okay, so let's just do a quick summary, and then we will also have time for questions. The skeletal, if we wanted to just summarize everything that we have gone over so far, just real briefly, the skeletal muscle is stimulated by a nerve impulse, which depolarizes the muscle. And we already know that not all muscle fibers will be activated at one time. So it depends on how much force is needed, which ones are activated. And the calcium ions are released from inside the sarcoplasmic reticulum into the sarcoplasm, your muscle, when it is stimulated. The calcium ions bind to a protein, troponin, on the thin filaments. This allows the myosin to bind to the actin. This action makes the thick and thin filaments slide past each other, shortening the muscle. And then that is a contraction. These are the works that were cited for this PowerPoint.